Well, nice to have all of you guys here. Thanks to Karen for including me today for this session. You guys should feel free to speak up anytime if you have a question. Interrupt me, it's not a problem. I can jump right back into the slides where we leave off. Really, my goal today is to kind of just give you a background of what I do at Aracari. So that's, we're a drug discovery company. We do, and I'm going to tell you about our platform and how we work with big pharma companies to work on discovering some drugs. I'll tell you a little bit about how I got where I am. It's not a roadmap by any stretch. Everybody's path is different, even though there's some small similarities. So let's go ahead and get started. Oh, slides. Okay. So I'm going to cover today how I kind of migrated from academia to a biotech startup. It's probably a traditional way for most scientists to get into the biotech startup space. But as I said, everybody's path is a little different. We all research different things. And that's certainly been the case for me. Just a disclaimer, because I do need to say it, everything I say today is my opinion. It is not does not reflect any of the opinions of Aracari Biosciences. So I just need to get that out of the way. All right, so we're going to start off. I'm going to put you guys on the spot. What is the first thing you think about when you think about a career in science? Don't be shy. There are no wrong answers.
Personally speaking, I think of like discovery and innovation.
Okay. Anybody else? Okay. So we'll get some shy people. There are going to be more points where you have to speak up, so just be ready.
We did have some responses in the chat box.
Ah, okay. So what do we have in the chat box?
We have trial and error, people in labs experimenting, doctors, medicine, question mark.
Okay.
Research and discovery, solving problems. That's it.
Yeah, so a lot of kind of getting in the lab and exploring new things is what I'm hearing there. And that's absolutely right. So my background is biology. So that's where I'm going to speak from today. So generally speaking, biologists study humans, right? That's the core of our question. We're asking questions, how do humans function normally? You know, how do you have a child that becomes a functioning adult? How do you get a small human become a bigger human? And we're also interested in what happens when we break. So diseases, there are a whole plethora of them out there. And, you know, something that people don't often think about, but with cancer, you know, this is the biggest disease that I think everybody thinks of right off the bat. And kind of the same molecular mechanisms that cause you to grow and develop normally are the same things that are hijacked and turned on inappropriately when you have cancer. So that's, for that reason, there's a lot of overlap in how we study just a normal human and a broken human. And a lot of what we're doing is trying to figure out how to devise some new drugs that might be able to intervene and keep people healthy and more normal. So there are some developmental defects where people don't grow up quite right or their organs don't develop quite right. So we might devise an intervention there. Same thing with diseases. Obviously, you're familiar with a lot of chemos and other types of interventions for cancers. So that's the big priority of what we emphasize as biologists. We're always trying to understand how all the cells and mechanisms work together to create a happy and healthy human. So when you think about drug discovery, I think most people think, where does research happen? It happens at research universities, so like UCI, UCLA, all the ones in the area. And that's not wrong, but it's actually only one part of the equation. So for example, there's a lot of really cool discoveries that happen in research universities, and not all of them will actually make it out of the academic labs into a place where patients can access them. So what we hope to find is that a really promising, maybe a lab professor has identified a new promising drug, and they're able to go spin it off into a small biotech company. Now, this isn't a one-way street. It's not like you just say, okay, I found a drug, now small biotech, you go take it away. There's actually a big partnership there. So small biotechs will often relay information back to the research universities or that specific lab. May partner with the research labs to figure out how do we further develop that drug, or maybe how do we develop related drugs. And of course, when we think about drug discovery, we also think of big pharma. And big pharma is often going to partner with small biotech because they have a lot more money and they have a lot more resources, right? So they can now take maybe a small biotech company that is developing one single drug, and they can now take it, test it in conjunction with a whole bunch of other drugs, and they have the money that can do very expensive clinical trials. I'm going to talk about that more in a second. But of course, the last edge of this ecosystem is that there is actually a lot of coordination between research universities and pharma. It's not very, I certainly wasn't aware of it when I was in high school and college, but it's something that I've become more aware of now, where big pharma companies will actually outsource projects that maybe need a little bit more liberty to explore interesting questions. Big pharma is going to explore things in a very specific way. Academia, you're going to go here and then find an interesting question and go over here. There's some real benefit to exploring some questions in a research environment versus a big pharma environment. So why should we have such a complicated ecosystem for developing drugs? It seems like there are a lot of players, and I'm just giving big categories here. I'm not talking about the individual biotech companies, the individual universities, and then the individual pharma companies. There are thousands of combinations when you think about it. The crux of the problem is developing drugs is very expensive and very time consuming. So the way we study drugs right now, this drug discovery phase, which may be done in all of those different entities in that ecosystem, they're screening thousands of compounds, and maybe they're going to find a few that are good. And now they're going to put them into traditionally mice in a preclinical trial and make sure they don't kill the mice or cause any adverse effects. And then only then are they going to go into clinical trials where they're going to go into a human, and then gradually you're going to get into testing efficacy and toxicity. Does it effectively do what it's supposed to do, and does it not damage the person that you're putting it in? At the end of the day, you are very likely to only get 1 out of 10,000 drugs that's successful. And it's going to take you 15 to 20 years. So I probably don't need to tell you guys much about this. This is, I'm sure, a very astute group. But when you hear about how expensive drugs are, this is why drugs are so expensive. You're paying for that one drug, but it's paying for those other 9,999 that failed. So that's partly why it's so expensive. So why do we have this problem of having such a low success rate? Well, it's really about how we study drugs. So cells are grown in a petri dish in the simplest of assays. It's simple. You have some, let's just take cancer cells, for example. You squirt some drug on them. Does it kill it? Does it not? Real easy to test that, and you can screen thousands of compounds at once. If we go into a little bit more complexity, we can put it in a mouse model. Let's say we have a tumor growing in there. We can put the drug in there, and that's good, but it costs more money, and it's maybe a little bit more difficult to interpret the results. And of course, what we really want is to go into human models. So this is typically clinical trials, which are very expensive and really can only be undertaken by the largest of small biotechs and big pharma companies. So there are a lot of cons to all of this. So in a petri dish, they obviously are representing cells that grow on a plastic dish, which I don't think anybody has plastic growing in their body where tumors are growing. So this isn't very representative of the body. Mouse models, I think for the most obvious reason, mice are not humans. And that's actually one of the key problems with mice. And then human models, you can't ethically put 10,000 different compounds in humans and have anybody be okay with that. You have to do all this testing beforehand before you put it in a human. So we came up with the idea a few years ago that we need a better way to study drugs in a 3D tissue, and that's more representative of what's actually going on in the human body. And so I'm speaking we because I was not involved in our initial efforts, but this is our academic lab at UCI. Actually had 3 labs that our company spun out of. And about 10 years ago, we looked at this very question: how do we make a better model? And we took a deep look at tissues in the body. And what do all tissues have? They have blood vessels. And blood vessels are important for not only providing the nutrients, but also taking away any waste from those cells. But perhaps most critically, at least for most drugs, they're delivered to your tissue of interest via blood vessels. So we felt that it was a very important component to have in any sort of model system that we were developing. And so over the course of 10 years, we've developed what I can show you here on the right is what we refer to as a vascularized microorgan. So it's got blood vessels that you can see in red. This is kind of a fluorescent dye that mimics blood. And in here, we can grow different types of tissues. So to give you guys an idea of the size, top to bottom of that chamber is about the width of a dime, the thickness of a dime. So it's very, very tiny. And we grow a whole bunch of these on one plate of cells. And it's 3D. So what you can see there, if you were to flip it around, it's about that thick. So it's very thin, and that makes it easier for us to image and kind of figure out exactly what's going on. So we put these cells in. So these are blood vessel forming cells. They're endothelial cells, and actually after only a few days, they'll actually form this very pretty vascular network here in green. And what's interesting is that we can actually put fluorescent dye beads through this, and you can see them flow through the vessels just like blood cells. So this is only a fraction of the data that we've produced over those 10 years, but we felt we had a pretty good model for vascularized microorgan. That's what we refer to it as. So if I may refer to it as a VMO, now you'll know what I'm talking about. All right, so let's get on to the career aspect of this. Let's step back. So how do you navigate to a career in biotechnology? What I'm going to tell you about today is my personal journey. And as I mentioned to you guys at the top, it's not necessarily the be-all, end-all way to do it. So pick up some tips where you can, but you don't have to do it the same way that I've done it. I hope you don't, actually. So just about everybody's biotech career begins at some point in academic research. So some people may do some work when they're in college, some research work in college, and decide they really like it. Some people may go on and do a master's degree. It gets them a little more experience in the lab, and then they may take a career from there. Others, like myself, decided to go for a Ph.D., which is about 5 or 6 years of very dedicated research. And then after that, you do more training, and then hopefully you have a career after that. So I have colleagues that have done every one of those, undergrad, master's, and PhD. They're all valid ways to get to a biotech career. It really just depends on where you want to go in the long run. All right, so as I mentioned, it's not a single path. So let me walk you through what I did. And I think you'll figure out it's a very circuitous path to start with. So I'm sure you guys are familiar with the central dogma of biology: DNA makes RNA, makes protein. And when I was an undergraduate researcher, I started working in RNA, which is very small. You can't see it. So you have to kind of figure out some different ways to look at other things. Look at them in a way that might give you a semblance about how they, a representation, I guess, of how they actually operate. So we were interested in how they actually fold onto each other, and that actually has some interesting implications in how you make a protein. So very interesting. I enjoyed it for the 2 years that I did it, but I said, that's nice, it's too small. I want to see something that's a little bit bigger, so maybe a cell. So also while I was an undergrad, I had a chance to intern at a company called STEM Life. And STEM Life worked on blood stem cells. And so they would take umbilical cord blood after a baby is born, it'd isolate all the stem cells, and then you could freeze those down and potentially take those later on and put them back into a person and maybe heal them of some diseases like lymphoma, leukemia, things like that. And I thought this was so cool. I'd never heard about stem cells before. This is a long time ago when before stem cells really took off. And I thought it was really cool. Thought it'd be really interesting to study more research in this. What was also cool about this was STEM Life was a small biotech startup. It was about 40 employees by the time I started. which is still considered a small company. I loved that you could just get your hands dirty on absolutely every aspect of the business, especially because I was coming in there with no experience. I had a couple of years of college under my belt, and that was about it. And it was really cool to get some exposure to that. All right, so I continued on. I actually ended up doing a master's and dabbling more in that RNA biology before I said, okay, that's enough, no more. And then I went off for a PhD. And in there, I actually decided to look at how the brain developed. So I was working with mice, and this was looking at stem cells in the brain. And so I would take the stem cells, grow them up, and actually look and see they form all the different cell types in the brain. And we were trying to see, do they form maybe a neuron more often than an astrocyte, which is a cell that supports neurons. We found some interesting questions there. I'm not going to go into the details because they're not that exciting for this. But it was really cool to kind of work at more of an organism level, a cell and organism level. And it got me to start thinking about how do we apply really cool discoveries in the lab to something that's more commercial. So at this point, I finished up my PhD and I started looking for applications. And that actually, that's when I started here at UC Irvine about 6 years ago. And I started working on a model of type 1 diabetes using our VMO platform. So in diabetes, you guys may have heard of pancreatic islets, and specifically beta cells. They produce the insulin in your body. And when you don't have insulin, your blood sugar goes through the roof and causes all sorts of complications. So people with diabetes will often have to take insulin by a shot to maintain their blood glucose levels at a normal level. What we've done is we're actually trying to study that in an environment. So that pancreatic islet, which you can see here in the purple cluster, we're actually trying to study it in the lab in a way that is closer to how they would be in the human, not necessarily how they would be in a mouse, because that's, again, mouse is not a human. So we tried to study this in the context of all these human cells. So as I said, it was about 5 or 6 years ago. And throughout this process, I've actually been working with my boss, and we always knew we wanted to take this VMO technology into a company. And so a couple of years ago, we started doing the groundwork for it. We had a patent from the beginning. And we started working on licensing that and actually putting together a team to build out a company. And so that's where we are now. We actually have a company. So how do we do that? How do we actually go from this cool idea in the lab that we've studied a lot and to actually get it to a startup? Well, in Aracari's case, we were founded in 2019. We ended up hiring myself and another colleague from my academic lab. Both of us had a lot of experience developing the platform. And we started working in the lab, and then we started working on what can we do to improve the platform. So we took this blood vessel, and well, we've done things where we've put various tumor types in there. So everything you can kind of think of: blood, colon, breast cancer, colon, skin cancer. We're working on some brain cancer now. And actually these tumors grow just like they grow in the body. So you can see it here in blue in the picture on the left. Because we have blood vessels, what also flows through blood vessels besides nutrients? Leukocytes or white blood cells. So we started putting those in, and we're doing some initial studies on how those might interact with the blood vessels themselves, or maybe also attack tumors or other types of tissues in there. So where do we fit in that drug discovery ecosystem? Well, I told you we kind of developed ourselves in the research university. We spun out to now this small biotech space about a year and a half ago. And actually our customer base is the big pharma companies. So we work with a lot of them to kind of negotiate on what are the questions they want to ask, what are the questions we can answer with our platform. And they give us some drugs, we run it through our platform, and then we send them lots of beautiful data back. And they come back with hopefully some more follow-up questions. So we work with a lot of the kind of the pharmaceutical companies that you might have heard of. All right, so lastly, what does a lead scientist mean? What does that title mean? Well, our company is right now, I think we have 3 lab employees, and we have a couple leadership team employees. So we have, I think, 5 or 6 employees overall, which means no 2 days are the like. Every day I'm doing something different, which I really like. So a lot of the days I'm doing research in the lab, working on those projects that our pharma companies have been interested in. We got to complete those contracts and send them back their data. Oftentimes I'm also working in the business development side. So this is actually networking and selling directly to our customers. So that's conference calls, that's going to conferences and networking there. I also work a lot on devising how we price this platform and the service that we provide to our clients. And then I also work pretty closely with these big pharma companies in coordination with some others on our leadership team to really devise the projects that we're going to ask the questions for them. And lastly, just kind of day-to-day operations. So there's a lot of project planning. We have 7 projects going right now. We don't have 7 people. So how do we stagger them all and make sure the Lego pieces all fit together and we keep everybody happy? So that's kind of that's 1 day a week that we're doing that. We obviously are expanding right now, hiring and training new employees. It's so critical to making sure that everybody's doing really good quality work. And then we're also developing strategies to kind of bring new products to the market. So other tumor lines, we have a blood-brain barrier model that we're still working on bringing to market. So those are kind of all areas that we're working on. So open it back up for a more interactive section. What do you guys think are the skills you really need to be successful in a career in a small biotech company? Anyone?
Flexibility, drive, patience.
No, that's a good one.
Enjoying the work you do. Oh boy. Collaboration, research, mindfulness, compassion, dedication, business. I might have lost it, but those are all really good thoughts, and I completely agree with them. So I put down 5. All of those are valid as well. So I'm going to focus on kind of soft skills. So one of them, absolutely stay curious and ask a lot of questions. When I'm hiring new employees, it's the #1 thing I look for in interviews. Are they curious? Are they asking a lot of questions? Are they really trying to figure out not only what our company does, but how they might fit with the company? So that's my best advice to you guys, to just make sure you're always showing that when it comes time to interview for jobs. Be a team player. I can't overstate this enough. When you have a small team at a small biotech company, it's so critical that everybody gets along. You don't have to be best friends, but you do have to play nice with everybody. So it's really critical that you're ready to be a team player. But that said, don't be afraid to lead. If you see something, this is especially true at small biotech companies. If you see something that is an area that the company should explore, go out and explore it and talk to people and say, hey, this would be really interesting to explore a little bit more. It's such a critical skill, and it's not something that you learn right off the bat. I think you get it kind of as you go along and get more experience. And lastly, communicating. I think one of the things they said was collaboration, same sort of idea. You want to communicate effectively with your colleagues. And you also want to be able to communicate effectively with people who aren't in your company. You want to be able to tell other people simply what you do at your company. So the biggest test for me is always telling my parents, and if they can follow what I'm saying, then that's pretty good, because they don't have a science background, either one of them. So if they can understand it and get as excited as I am about it, then we're winning. So that's something to kind of think about. And those are skills that I would suggest that you guys continue to develop as you go along. This workshop, this couple of weeks, is doing a lot. I know you guys are getting some experience in pitching next week. So you guys are going to nail that communication skill. So with that, I'd like to just open it up for any questions that people have.
Okay, we have one question from Rami. He asks, you stated previously that drugs take 15 to 20 years to be made. Is this going to be the same for vaccines like for COVID-19?
Yeah, it's a great question. I think we don't really know right now. There's a lot of effort that's trying to kind of shortcut that pipeline that I showed. So we're going to cut out some pieces there that may not be as critical so that we can get a vaccine done, hopefully in the next year, I think is the goal. It won't take 15 or 20 years. We're going to make sure that that doesn't happen. But it will take some time, and we're doing something that we've never really done before by trying to make a vaccine so quickly. It's a great question.
Hi, Dr. Bender. I have a question about the clinical trial slide that you brought up. I
Sure.
see that you showed humans as a subject for clinical trials. So I know you all, or not you specifically, but researchers can also use monkeys or other like non-human primates. So
Yeah.
how do you determine which is used in a clinical trial?
Yeah, so I think everybody strives to use human because it's going to be the closest to where the drugs are ultimately going to be used. So monkey and non-human primate trials have actually been minimized in the last 12 months. There's a very concerted effort to kind of go away from them. In some ways, kind of like mouse studies, we try to minimize how much we do animal studies. So they are less utilized at this point. The goal really is to get it into humans as soon as possible, though, because what might be toxic to a human might not be toxic to a monkey. And that's really important to figure out. There's kind of no way to figure that out until you put it in a human.
We have a couple more questions in the chat. Andres would like to know, are there any resources to find lab work, internships, and other opportunities for someone interested in gaining experience in lab work as a high schooler?
Yeah, that's a great question. I don't know of dedicated websites or resources like that. But you know what, we've had several people come through as high school researchers in our academic lab. I would encourage you just to start looking at labs that you think are interesting at your local university, whether it's UCI or somewhere else. Start looking at labs, find out if there's an interesting question, and there's no reason that you shouldn't reach out to a professor and say, hey, I'm really interested. Is there a possibility I can intern? They won't advertise a lot of those positions, and professors are busy. So spoken from experience, be aware that most of them won't respond to you. Don't be disheartened about that. They just get a lot of emails. So feel free to follow up with them again, and then and look at others. There is, what I hope you take away is that there's no right way to get research experience. You don't have to find research that's going to be what your career is right now. Go get some research experience, get your foot in the door. I think it's great. But don't be afraid to just reach out to professors.
Great, I can add to that. I can add to that too. Dr. Bender, DLP is offering a hands-on lab course training in August and September. So there may be internships tied to that after the successful completion of the course with some of our resident companies here. So please go ahead and online, and I think we've already distributed that to the students. You can apply. There are financial need scholarships. So please take a look, and that's one avenue to skill up first, and then try to find undergrad opportunities.
And thank you, Jessica. She just sent a link for biology-related internships for high school students.
Great.
So click on those links, Andres. And we have another question from Ko. He's asking, is there anything that you did in terms of your education or career decisions that you're glad you did and that helped you get where you are today?
Yeah, I think the biggest thing that I'm really glad I did is I didn't go straight from graduating college into a PhD program. So I've talked to a lot of my colleagues about this. I certainly know some people that went straight away, my wife included. But I think there's a big benefit to do what I do, where I did, which is go do a master's. You get some research experience in a slightly less intense environment, but you can really build up some skills that are going to help you a lot in your PhD. I think it made my PhD a lot easier because of that. Even if you can't do a master's or you don't want to do a master's, go do some work in a biotech company, an internship for a year or two. I wouldn't say much longer than a year or two. A year or two is great. Like, figure out if you really like the science and the research aspects, and then if you decide to go to a PhD, then great. If it's not your thing, like I said, there's no problem with that. But I think that was, I think that for me, that was the key, because I hadn't zeroed in on enough on what I wanted to do right out of college.